Hello and welcome to Medicine News Daily. Here today we have with us Dr. Taylor Gerke. I'm glad to have you here.
Thanks, it's a pleasure to be here.
If you guys didn't know, Dr. Taylor Gerke is a very highly respected virologist.
Correct,
virologist in her field. So, how did you get started as a virologist?
So yeah, my education consists of graduating from high school with an interest in microbiology, biomedical sciences, and epidemiology. I got my bachelor's in biochemistry at UW-Madison and then transferred to Emory University for my graduate where I got my PhD. Then I did some postdoctoral research training for around 4 years through the NIH Intramural Research Program. Through all of this education, I make around $77,000 a year by preparing, conducting, and overseeing studies of microorganisms and research studies with scientific equipment and labs. So, if you don't mind, I'll give a little bit more information on my day-to-day stuff for any aspiring virologists out there.
That'd be good.
So I observe bacteria that's become host to a virus by applying different levels of moisture, temperatures, and air conditions to see how it reacts.
Mm-hmm.
I also am more recently been supervising a team of researchers in university settings or larger private lab facilities. And in this, I coordinate and supervise the research process as opposed to actively planning it or participating in it. Once research projects are completed, I communicate results to my employer. My technical reports and presentations may go to other researchers, lab administrators, or government health agencies. At the highest level, virology, the field of science I'm in, contributes to plans developed by the Federal Center of Disease Control, or CDC, to control outbreaks of influenza and other viral infections.
Wow, thanks for that overview. So what is this thing that you've developed?
I'd love to, explain about that. So my colleagues and team have been hard at work developing a vaccine for the Epstein-Barr virus, or EBV, which is a virus that before us and a few other small teams have been working on. There are no published medications or vaccines for it at this time, so it's high demand.
Yeah.
EBV, in case anyone doesn't know, is a type of herpes virus known for causing infectious mononucleosis, or as it's commonly known, mono. EBV infects and stays in B lymphocytes and epithelial cells. As for any other virus, it's important to have vaccines in order to help people's immune systems have a better chance at defending the body against the virus. But it's especially important that EBV has a vaccine, which is what we're working on, as it's known to increase a person's risk of developing nasopharyngeal cancer and Burkitt lymphoma. EBV is also linked to Hodgkin diseases and stomach cancer.
Wow, that sounds really serious.
It is.
Would you be able to tell us more about this vaccine?
Of course. So the vaccine isn't completely ready for mass market yet, but it's almost there. We've successfully tested the vaccination on mice and monkeys and are on the verge of starting the stage of clinical trials with people.
Huh, that sounds really promising.
It is. Like I said, we're mostly excited about the breakthrough that allowed for successful testing on animals and the potential it holds for prevention for cancer for millions of people.
Yeah, it sounds really great. Would you be able to give us a more in-depth description on the vaccine you're developing and how it will be able to prevent cancer?
Of course. Prior efforts to develop an EBV vaccine focused on a viral surface protein, GP350, that the virus uses to enter B cells. However, EBV infects not only B cells but also epithelial cells that line the mouth and upper throat. These cells are usually infected after contact with saliva from an EBV-infected individual. The new research that we're working on helps define the contributions of virus-neutralizing antibodies other than those directed at GP350 on B cells. Among other findings, our team has determined that antibodies to viral proteins called the GH/GL complex play a major role in inhibiting GBV fusion with epithelial cells. Our group has also developed 2 vaccine candidates, so 2 versions of this vaccine, one designed to elicit antibodies to GH/GL on epithelial cells, and another that included GH/GL and another viral protein GP42. In both candidates, Each of the experimental vaccines included antibodies that potently inhibited epithelial cell fusion. The vaccine containing GP42 induced stronger B cell fusion inhibitory antibodies than the one containing GH/GL alone. Unlike the GP350 candidate EBV vaccine, which protects only B cells from infection, the candidate vaccines described in our report on this new vaccine that we're producing and trying to research elicited antibodies that could prevent EBV from fusing with both epithelial cells and B cells and may provide protection independent of cell type. Sorry, it's a lot. It's kind of technical.
No, I think that's really good for listeners who are interested in this stuff.
Yeah.
Earlier you mentioned working with a team of people. So what is your company that you work called, and what is the name of the product that you've been developing?
Yeah, I work for NIH's National Institute of Allergy and Infectious Diseases, NIAID, and the name of the product is simply called EBV vaccine.
Well, I'm really glad you can make this to this interview we have here. I'm sure our audience really enjoyed and understand some of this breaking news in the virus and cancer field.
Of course. Thanks for having me. Now, if you don't mind, I have to get back to my lab. We have a lot of work to do with viruses.
I don't want to keep you from that.
Thanks. Thanks for having me, and have a good day.
And that was Medicine News Daily. Please come back next time for how you can cure cancer and corona in 12 easy steps. See you next week.